Okay, we're going.
All right. Okay, well, let's take a look at these processes. The major things of RNA processing: capping, polyadenylation, splicing. Now, the capping, like I say, different types of RNAs have different caps. Some have none at all. Messenger RNAs have a specific cap. Polyadenylation, that's unique to the poly A tail, that's unique to messenger RNAs. Okay, so let's take a look at this. But before we do that, let's take a look at this thing about why RNA processing can be important.
What's the first word?
Oh, capping.
Capping and then
Polyadenylation. Yeah. Okay, well, let's first take a little look at this kind of aspect. Why do we do this? Okay, couple interesting little experiments that were done. And this is hybridization experiments. Now, let's use as an example the Globin genes. These are the genes that make the protein components of hemoglobin. So you have an alpha globin, 2 copies of that, and a beta globin. That makes up hemoglobin when you throw the iron-containing heme groups in there. Okay, so these are the proteins that make up hemoglobin. Now, the only cells in an organism that actually produce hemoglobin are the red blood cell precursors. Now, in mammals, what happens is these precursors, called erythroblasts, what's going to happen with these guys is as they go through the final stage of development, they lose their nucleus. That means they can't produce anything anymore. And a mature red blood cell has no nucleus, and it's basically a cell membrane with receptors and a cytoskeleton underneath, and then the inside of it's almost all hemoglobin. Now, not surprisingly, since it has no nucleus, mature red blood cells cannot divide, nor can they produce anything new. So their precursors, the erythroblasts, have to do all that stuff. And red blood cells in the human body last only about 3 months before they die off, and they have to get replaced. You're constantly replacing red blood cells as they die off. Okay, well, the only cells that are producing and expressing the Globin genes are going to be these erythroblasts, the red blood cell precursors. Now let's do a little experiment with hybridization. We're going to look for Globin RNAs. So let's do this little experiment here, hybridization experiment. We're going to do 2 things. We're going to isolate the messenger RNA, total messenger RNA, from a couple different cell types. And we'll put over here RBC, that's red blood cell, actually precursors. Over here, some other kind of cell, let's say liver, and let's say skin, for instance. And this is going to be messenger RNA. So we make probes to these Globin messenger RNAs. And now what we do is we're going to hybridize with total messenger RNA, which is what we've run on the gel. Okay, and we hybridize with these probes to Globin messenger RNA. So, if there happens to be Globin messenger RNA around in the cell, you're going to see it with the hybridization. And you'll ignore everything else, all the other messenger RNAs. Okay, well, we look at the red blood cells, and not surprisingly, there is a gigantic amount of Globin messenger RNA, at least the red blood cell precursors. We look at liver and skin cells or other cell types, and we see nothing. That's telling us that the Globin messenger RNA is unique to do the red blood cell precursors. But now let's do another experiment. We're going to still use our probes to the Globin messenger RNA, but this time we're going to extract nuclear RNA from different cell types. The RNA in the nucleus. Isolate the nuclei, break them open, and then collect all the RNAs. So, now we have nuclear RNA on this side. Okay, so let's do the same thing again. Our red blood cell precursors, say liver, and say skin. Okay, now, not surprisingly, huge amounts of globin nuclear RNA, that's the messenger RNA precursors, are found in the red blood cell precursors. Okay, huge amounts here. Let me see if I turn this just a little bit. Just a little more visible. Okay, let's see how that works. Hang on. Oh, it's hard to say when I actually apply video. Okay, now, we do this with liver and skin. When we looked at messenger RNA, we saw absolutely nothing. But when we look at nuclear RNA, we get a different story. We see a little bit of globin nuclear RNA. Not much compared to what we see here, but we do see a little bit. That's telling us something very important. That's telling us, to some extent, the globin genes in other cell types are being transcribed. Now, granted, they're not being transcribed in huge amounts, but they are being transcribed a little bit. But those RNAs never make it out into the cytoplasm. We don't see a trace of globin messenger RNAs in other cell types. Now, if you do similar hybridization experiments, you get total messenger RNA from one cell type and from another. Let's do another experiment, a little mental experiment here. We're going to have probes. We're going to make probes to let's say from liver cells. Okay. Now, we're going to look at messenger RNA from different cell types. And we're going to see how much hybridization. Oops, sorry, liver. Let's say skin, kidney. It could be any cell types. It's just totally arbitrary. We do, using probes against liver total messenger RNA, of course, we see a huge amount of hybridization. In fact, it's about 100% hybridization. Not surprising, right? Because all you're doing is probes against the very same messenger RNAs that these liver cells produce. Okay, now when we do that with skin or some other tissue, we see some overlapping, we see some hybridization, but nowhere near as much as we saw in the case with liver. So let's say about 20% and maybe about 30% for here. Now this is telling you at the level of messenger RNA, what we see here is there is a partial overlap between the messenger RNA that's in liver cells and with other cell types. Maybe 20–30% is typical. But there's a lot of messenger RNAs produced in liver cells that we don't see in other cell types. So what we're seeing here essentially are the genes that are going to be expressed in almost all cell types. These would be the so-called housekeeping genes. These are the things these housekeeping genes are basically the genes that code for proteins that are fundamentally important for cell survival. Some cytoskeletal proteins, certain receptors, the enzymes of metabolism, the proteins of ribosomes, DNA replication and repair enzymes, that type of stuff. Stuff that you absolutely need for the cells to survive. So that's saying that all these cells share a certain percentage of expressed genes in common. And those are the genes that are necessary to keep a cell going. But that there are significant differences. There are a lot in a specialized cell type. There are a lot of messenger RNAs that you're not finding in other cell types. Those are the specific ones. Those are the RNAs and the proteins they code for that make a liver cell a liver cell. And of course, if you did the same thing with skin or kidney or whatever, you'd see the same kind of thing. You would see that there are certain genes expressed in common by all cells, but there would be a lot of unique genes. How many depends on the cell type? For instance, the red blood cell precursors, there's only a handful of unique genes in those cells that are being expressed. On the other hand, you look at things like, say, neuron in the central nervous system, there's about 10,000 specific genes being expressed. So it depends on cell type. Okay, so we do this experiment. Now, forget the messenger RNA. Let's do, here again, probes the total liver messenger RNA. But now we're going to hybridize against nuclear RNA. All the stuff that's in the nucleus, including precursors and what have you. So we'll look at total nuclear RNA. And when we do that, we see a radically different story. In all cases, the hybridization is close to 100%. This is telling us something again, just like what we saw with this experiment here. This is telling you that virtually all the genes that were expressed in the liver cells are being transcribed into nuclear RNA, at least at low levels. But only certain of those RNAs actually make it out of the nucleus as mature messenger RNAs. So here we're seeing, this is a global view, this is a view of a specific RNA. We're seeing something very important here. This explains some reasons, one of the reasons why we do this RNA processing to begin with. Is it okay if I erase these diagrams? Okay. All right, the take-home lesson of this is this. Okay, point number one. Virtually all genes in all cells, at least all the protein coding genes, are going to be transcribed to a limited extent. Even in cells that are not supposed to be transcribing these genes. Liver cells and nerve cells are not supposed to be transcribing the globin genes. They're never going to use globins. But they do. Not huge amounts, but they transcribe some of them. Now, second important point here. Only certain of these nuclear RNAs are going to get processed. Only the right ones are going to get processed completely into messenger RNAs and go out into the cytoplasm. Okay. Now, why does that happen and why is it important? Well, you know, it's kind of like the old thing. Many are called, but few are chosen. In this case, many are transcribed. In fact, virtually all of them are being transcribed, but only a limited number are chosen for processing into the mature RNAs. You guys have questions?
Yes. Is there supposed to be two ats? Like,
To what?
It's supposed to be
AT?
at least.
Oh.
Nearly all genes are
At
transcribed.
least
It's supposed to be
low
low.
levels are transcribed at least at low levels. Okay, at least at low levels. Okay, that's oh, I just put that too high. That's what happened. Okay. Now, why do we get mistranscription of genes, right? We said that a whole big thing of regulating transcription is that different cells are going to have different specific transcription factors, and they're going to transcribe certain genes and block transcription of others. But what happens is when you form a basal complex, here's our promoter. Okay, basal complexes, in order for transcription to take place, have to assemble together. You have to assemble together the polymerase and all these basal transcription factors and things like that. And they have to hold together long enough for transcription to take place. Now, ordinarily, a basal complex may form or partially form, and then it will fall apart. Something will fall off, and the whole thing will fall apart before transcription can actually happen. And that's generally the case. Now, these specific transcription factors, at least the ones that are activating transcription, what they will do is they will stabilize the basal complex, making it more likely to hold together long enough for transcription to happen. But when you come down to it, it's a random thing. You could say that a basal complex has, let's suppose it has to hold together for 1 minute for transcription to take place. You could say that in the absence of a specific transcription factor on a given gene, a basal complex has maybe a 1 out of 10 chance of holding together long enough for transcription. Add the specific transcription factor, maybe that's a 90% chance. But it's still a roll of the dice. So once in a while, on a gene that you don't want transcribed, a basal complex will assemble, and it actually, just random luck, holds together long enough to activate transcription of that gene. It may not be likely, but it can happen once in a while. So that's how you get mistranscription of genes. Just a roll of the dice, and the cell has rolled snake eyes. It's transcribing genes it doesn't really want to transcribe. Okay, now, if you're a bacteria, you don't have all this RNA processing. If you accidentally transcribe, quote, the wrong gene, you know, like a gene that codes for an enzyme that can break down a food source that's not present, okay, what has happened? Big deal. You wasted some amino acids, you wasted some energy to make a protein you don't need, but it's not really going to be that much of a disaster for you. It's a little bit of a waste, but okay, well, what the heck. On the other hand, if you're dealing with in a multicellular organism that's capable of development, if you mistranscribe the wrong gene, that gene may code for a transcription factor that can activate a complex developmental program. To give you an example, the ILIS or PAX6 gene. Suppose it accidentally gets transcribed in your butt. Do you really want the cells that are coming out of your butt? Do you really want eyes right here? Probably not. But that could happen. Recall those fruit flies with eyes on the knees? Okay, oops, I'm sorry, we just turned on PAX6 here. It's going to look really weird when you take your pants off. Oh my God, I can see out here. What? Okay, you don't want that to happen, folks. You don't want to end up producing proteins intended for another cell type or building structures that you're not supposed to be building in that part of the embryo. So in this case, in a developing organism, if you transcribe the wrong gene, the results could be disastrous. You could start off a chain reaction that could completely affect the differentiation of that cell or that tissue, having it turn into something else that you don't want, or at least you don't want it there, like eyes in your butt, or having a weird hybrid that has characteristics of two radically different cell types. Okay, that's bad. So, what happens when you make a mistake here and mistranscribe a gene? Accidents do happen. We have a backup mechanism. In the nucleus, only the genes you want transcribed are going to be processed into messenger RNAs. The accidents never get processed. Therefore, they stay in the nucleus and eventually the nucleases get to them. So in other words, you screw up, cover up your mistake, shove it under the rug, let it bury it. Nothing's wrong, don't worry. Sounds like a lot of politicians, right? They screw something up, oh, we didn't do anything, you know. Okay, until a congressional hearing comes along. But this is the kind of thing that happens. You make a mistake, you have the opportunity to basically cover it up, and like it never even happened. The cytoplasm never knows that that gene was being mistranscribed. It's never going to see the messenger RNA, so the protein's not going to be produced. So this decision about do we or don't we process an RNA is a backup for mistakes in transcription. Yes?
So if it's supposed to catch it, why was there flies with eyes on their knees?
Oh, that case they did it, or it was deliberately done artificially, where they expressed everything they needed. They deliberately expressed. In that case, it was sticking a powerful enhancer for another gene on there that was present and that transcription factor was present in those cell types. They linked it, so they took that eyeless gene, stuck in the knee, and attached with molecular biology, gene splicing techniques, attach an enhancer that was supposed to be expressed there anyway. And yeah, So that was a nasty little genetic engineering trick to see whether that would actually work, whether that really did lead to the development of an insect compound on it. So yeah, that was an intentional kind of thing. Okay, so all right. So we can cover up our mistakes that way, which is actually very important in development. So that's one big type of thing about why we process RNAs. Yeah.
So if RNA processing is used for, getting rid of the genes that aren't supposed to be translated, then why does the cell just keep transcribing genes, all the genes?
It's just simply random mistakes. Whether a gene gets transcribed or not is going to depend on whether a basal complex can form there and hold together long enough. That's a basal complex. Let's suppose something. Have you ever heard the concept of a half-life of something? Okay, we could say here, okay, let's suppose in a given gene, in the absence of any specific transcription factor, the gene will generally not be transcribed. Okay, what it means is as we assemble a basal complex, components may shake themselves off and things like that. To have an intact basal complex, how long does an intact complex last? Okay, let's say 150. Okay, let's suppose most of the time a basal complex is there for only a few seconds before it falls apart. Not enough time for transcription to take place. And in, say, 30 seconds, half of the ones that are formed have already fallen apart. Transcription still has not taken place because it wasn't together long enough. Okay, and then, say, a minute, only a quarter is left. And so on. So we could say half of the formed basal complexes last only 30 seconds. But it doesn't mean that once in a while, let's suppose you have to hold it together 2 minutes for the threshold. I don't know how accurate that is, by the way, but in case of an example here. Okay, well over here, by the time you get to 2 minutes, only maybe 1 out of 10 or 1 out of 20 basal complexes will have held together that long. Now you can't say, looking at a newly formed basal complex, you can't say you're going to be there for 2 minutes, for 3 minutes, and transcription will happen, but you, the same basal complex, you might be there for 5 seconds. I can't tell. All I can say is there is a tiny probability that you'll be there, and a much higher probability that you're going to fall off. So it's a random roll of the dice, in effect. And that's what happens. Now, if you have a specific transcription factor that stabilizes the basal complex, then its period of stability is much longer and transcription is much more likely to take place. Did that help out?
Yes.
That's a good question.
It said you said just nearly all genes, and I just don't understand why it's why is it making so many mistakes?
Oh, well, it's just that
good at not making mistakes.
Well, yeah, but it's just that assembling and holding together a basal complex is kind of a random roll of the dice. How long it will hold together is a random thing, because all these proteins, don't forget, proteins don't look like those nice, beautiful, static tertiary structures you see in textbooks and journal articles. They're actually wiggling around quite a bit. Now let's suppose, all right, I got an idea. Let's suppose I'm part of a basal complex. This is one of the proteins that's part of the basal complex. Okay, now, I'm a molecule. I'm in constant motion. I'm wiggling around and jiggling around, and I just knocked a critical component of the basal complex off. Now, that took about 5 seconds. Okay, all right. Okay, so we're going to say 5 seconds. Now, let's see how long it takes now. It's still assembled, and I'm wiggling and jiggling around, and I'm wiggling and jiggling around. This ain't coming off yet. Okay. All right. Now that was about 10 or 15 seconds. I cannot predict when I put this thing on, is it going to be on me for 1 second, 10 seconds, or a whole minute? If I do this for a minute, I'm going to get damn tired. But, okay, because I can't predict. Oh, that was about 3 seconds. You see how that goes? It's a random process. So, you can up the odds of it holding together by stabilizing with a transcription factor or factors. You can reduce the odds by disrupting a transcription factor. So if I stabilize it, for instance, maybe I take a little bit of glue or something and glue it to my head, and then it can hold on to me for a long time. If I destabilize it, maybe I put real slick, greasy stuff here, or put slick, greasy stuff in my hair. No, I'm not going to do that. And the chances of it sliding off will be a lot greater. Okay, did that help explain that? Okay, so yeah, accidents do happen because it's really how long one of these things hold together is really ultimately a random process. Okay. All right, no more basal complexes for a while. All right.